Okay, thanks for your response. Okay, so we have another classification of carbohydrate, which is disaccharide. So di is referring to two combination of saccharide, and monosaccharide is mono. Mono is just only one saccharide. Disaccharide is a combination of two saccharide. Okay, disaccharide consists of two monosaccharide. Monosaccharide links link together with glycosidic bonds in the alpha or beta orientation. So here you can see this is glycosidic bonds. There is actually two monosaccharide. Okay, this is sucrose lah. Sucrose is one of the disaccharide. There is one monosaccharide attached to another monosaccharide with the glycosidic bonds in the form of alpha and also beta orientation. There is actually different position alpha and also beta orientation. You will learn after this. Okay, this lactose is combination of two saccharide, okay, with the glycosidic bonds. So this is what we call it as glycosidic bonds now. Glycosidic bonds and also glycosidic bonds. So the most important of the disaccharide is sucrose, lactose, and also maltose. Sucrose is the most abundant and consists of a molecule of alpha glucose and also beta fructose. Okay, there is also alpha and also beta. And glucose, this is glucose and also fructose. So this is actually different form of structural molecule. The conformation, they call it as conformation. Okay, in the form of ring, still in the form of ring. Okay. This is also in the form of ring. Okay, alpha glucose, also beta fructose. This is the fructose lah. Fructose is a 5 ring. Okay, glucose to 6 ring. And lactose is found in milk and dairy products and consists of galactose and also glucose. Okay, here is the galactose, here is the glucose. And maltose is mainly produced by partial hydrolysis of starch. So starch is the polysaccharide. So it will be hydrolyzed into a disaccharide, and one of them is maltose lah, and consists of two glucose unit linked by alpha 1,4 glycosidic bond. Okay, I will show you what is alpha 1,4, beta, and so on lah. Some nutritional important disaccharides and disaccharide derivative, and their significance can be seen in this table. This sucrose is a constituent of fruits, vegetables, and so sweetener. Lactose is a milk, come from the milk and so dairy products. Okay, maltose and isomaltose also constituent of the starch. And there is actually other disaccharide, but not common saccharide lah. Okay, it's a trehalose and also lactulose. And trehalose is the food additive and constituent of mushroom. Lactulose is a lactose derivative and it's a laxative lah. And also there is a disaccharide alcohol, maltitol, and constituent of starch, which is also considered as sweetener, lactitol. And tol only meaning that it is an alcohol. Disaccharide contain alcohol. But you just need to understand sucrose, lactose, and also maltose only. Okay, first is sucrose. Sucrose is also known as table sugar, or just only sugar lah, in sucrose. Sucrose ni is naturally occurring sugar found in various amounts in plants, like fruits, vegetables, and nuts. And also is produced commercially from sugarcane, also sugar beet. So what you eat actually is a sucrose lah. And when we consume sucrose, it is broken down into equal part of glucose and fructose. So this is the structure. The hydrolysis of this disaccharide and disaccharide sucrose here will produce glucose and also fructose. Okay, glucose and then once it's broken down to glucose and fructose, and the glucose here taken up by our cell with the help of insulin, and while fructose is handled in the liver and does not need insulin to be absorbed. Regardless of its source, sucrose provides 4 calories per gram, and our bodies process in it in similar ways. Okay, so once we consume those fruits or vegetables or nuts that contain sucrose, this sucrose will be hydrolyzed into glucose, and then glucose will be taken up by our cell from the insulin. So that's why if those got problem with the insulin production, so you need to inject the insulin. Okay, but not for the fructose lah. Okay, fructose does not need insulin to be absorbed. Okay, if you see this the diagram here, okay, it's a combination of alpha D-glucose. You know that it is an isomer D-glucose. Alpha ni at this position, okay, alpha, and this is carbon number 1, and it will combine with the beta. Beta is in this form lah, H2OH, okay, and then at carbon number 2. Okay. So actually it's a combination of alpha D glucopyranosyl 1 and 2, beta D fructofuranose, we call it S-sucrose. So this is the long name lah for the sucrose. Okay, so it's a combination of glucose and fructose, and this sucrose will be will be broken down. Okay, will be broken down into glucose and fructose. Okay, another one is lactose. Okay, lactose is a combination of galactose and glucose. Same like sucrose. So lactose ni will be broken down into galactose and also glucose in our body lah, in the presence of the enzyme lactase. And lactose digestion occur in the small intestine with the help of the enzyme known as lactase. And lactase breaks down lactose into the monosaccharide galactose and also glucose, and then eventually this glucose will be absorbed by our body, similar to the sucrose lah. Okay, and then galactose is transported to the liver, where it is turned into glucose for use in energy production. So galactose also will change to the glucose, and then glucose in our body will produce energy. Okay, lactose is sometimes referred to milk sugar because it's only found naturally in the milk of mammals, including cow, goats, and human. Okay, milk from cows and goats is used to make cheese and yogurt, but not all milk products contain the same amount of lactose. Okay, so you can read from here lah. I'm talking about the hard cheese. Hard cheese are lower in lactose due to removal of whey and so on lah. Okay, so sugar ni, carbohydrate ni is not just come from the sugar that we know. Okay, it can also come from here, milk sugar. But there is actually what they call the reaction in our body that will degrade this 2 saccharide into 1 single saccharide, because eventually the glucose or galactose, finally glucose lah, mainly the product that actually taken up by our body, absorbed by the cells, and will produce energy. And the main thing is the glucose lah. Whether you eat those contain starch ke kan, contain polysaccharide ke and rice ke, so finally it will produce a glucose. So that's why glucose is very important. Okay, that's why we need to have an insulin and to control this glucose in our body. Okay, another disaccharide is a maltose. Okay, Maltose, you can see here, is a combination of glucose, two glucose. In maltose, the glycosidic oxygen atom of one glucose is alpha. Okay, so this is alpha 1,4 glycosidic bond. This actually alpha meaning that this comes from the alpha glucose here. Okay, and then this is beta. Ini beta glucose lah. And then this is at position number 1, and this is position number 4. Okay, 1, 2, 3, 4, carbon number 4. So that's why it's alpha. 1 referring to the carbon here, 4 prime referring to another glucose. This glucose comes from the beta glucose. Beta at this position, okay. And it's alpha is bonded to C4 atom another glucose unit that is like a glycol. So therefore, maltose is an alpha 1,4 glycoside. This is the other name for the maltose lah. Maltose is produced by the enzymatic hydrolysis of starch. Maltose is come from the starch. It's come from the polysaccharide, catalyzed by the enzyme amylase. And if you see the structure here, okay, the mechanism here, maltose, okay, will be react with the enzyme. Not react. It's not reaction actually. Okay, the enzyme will be taken up by maltose, and there is an active site for the maltose, and it will produce 2 molecule of glucose. Okay, maltose from the starch ni is further hydrolyzed by the enzyme maltase to produce 2 molecule of the glucose. Okay, the maltase is the enzyme. Is an enzyme that responsible for the production of glucose and by the maltose. The maltase enzyme is a protein that is perfectly shaped to accept maltose molecule and break the bond. Okay, the bond here. And the 2 glucose molecules are released. 2 glucose molecules are released. And a single maltase enzyme can break in excess of 1,000 maltose bonds per second and will only accept maltose glucose. Okay. So if you can see here, sucrose also contain glucose. Okay. Lactose also contain glucose. And finally, galactose will be converted into glucose. And maltose also will produce glucose. So glucose is very important. Okay, it's very important monosaccharide for our body. Okay, so this is what I told you about. The monosaccharide, the final monosaccharide is a glucose. This actually comes from the maltase, from the maltose, sucrose and lactose. Okay, so there is a little bit different here, and with other disaccharide. For the sucrose and also lactose, there are a specific enzyme that can broken down the sucrose and also lactose into monosaccharide. Okay, sucrose will have a sucrase to produce glucose and also fructose. For this one, lactose will produce glucose and also galactose with the help from the lactase. But for the maltose, it's actually come from the starch or glycogen, and this starch and glycogen with the help from the amylase. Amylase is an enzyme that can break this the bonding inside the starch and also glycogen. Will produce disaccharide maltose, and it will produce glucose with the help of maltase. Okay, so you can see the sequence here. It's come from the polysaccharide, will produce disaccharide, and finally monosaccharide. So actually the glucose that will be uptake by our body is important to our metabolism. Okay. This is what I told you about sugar. Okay. Sugar ni it can be divided into natural sugar or added sugar. So this actually is important when you want to eat. Okay, when you would want to select what is the suitable food want to eat. Okay, natural sugars come from the fruits, and example banana, mango, papaya lah. And added sugar tu you can get from soft drink, from candy, fruit drinks, bottle to pastries and so on lah. So that's actually added sugar. Okay. So natural means just got that from the natural lah, from the plant. Okay, from the fruit. The sugar is naturally part of the food. 100% fruit juice, honey, molasses, or milk. But added means that sugar was added during the processing, preparation, or at the table. It's not just only sugar we put it in the water, okay, in the coffee, but you can also see sugar this inside the soft drinks. Avoid foods that contain high fructose corn syrup, white sugar, brown sugar, corn syrup, and so on lah. They are all added sugar. Okay. Fruit pancake, syrup, fructose sweetener, and so on lah. Okay. So there is one question from your friend here. Doktor, kalau ambil banyak sugar dari buah, okay? Yep. Actually, you are not taking any sugar from the fruit. You just eat fruits. So actually no, there is no issue on that actually, and because in the fruits there are a lot of fructose. Yes, it's okay. Not we are not taking sugar from the fruit, but we need to eat a lot of fruits, and inside the fruits there is a natural sugar. Itu je. Okay. So if you eat a lot of fruits, they will not increase your level sugar, the sugar level in your body. It's not like that. Okay. For those who are diabetes, you can actually eat a lot of fruits. However, there are certain fruits that will give direct glucose to our body. For example, like watermelon, pembikai, and because it will give direct glucose lah to our body. And then other fruits, durian. Durian also will give higher sugar inside the body lah. Okay, so this also you need to know about the fruits, but most of the fruit is okay. Okay, there is actually term what we call it as reducing sugar. The type of sugar that acts as a reducing agent and effectively donate electron to some other molecule by oxidizing it. Actually, if you see here, D-glucose is a aldose. It contains aldehyde. D-mannose is contain aldehyde. Okay. Reducing sugar have a free aldehyde or ketone functional group. Okay, any sugar that tends to act as the reducing agent, since it has either aldehyde group or ketone group, is called as reducing sugar. A sugar that serves as a reducing agent due to its free aldehyde group. Example of the reducing sugar is glucose, fructose, lactose, arabinose, maltose, except for sucrose. Sucrose is not reducing sugar, and most of the monosaccharide and also disaccharide, they are reducing sugar except for sucrose. So what is a sucrose? You see here. Sucrose ni will produce sucrose in its form lah. Okay. So here is sucrose. Here is the sucrose structure. Okay. It's not considered as reducing sugar. Okay, uses of reducing sugar. Actually, you will learn about this reducing sugar in the biochemistry subject, and you will do this in the lab lah for the reducing sugar. Okay, we'll look at the use of reducing sugar Okay, in medicine, the Fehling solution has been used as a test to detect diabetes in human blood. Okay, like I told you about the diabetes, if you want to determine the sugar level in our blood, so we actually use the Fehling solution lah. The relative measurement of the number of oxidizing agent reduced by the available glucose makes it easy to calculate the concentration of glucose present in the human blood or urine. And because of the glucose too can be acting as a reducing agent to oxidize something. Macam tu. Okay. No, to reduce agent. It's a reducing agent for the reduction. Or it's a little bit complicated actually. The number of oxidizing. It is a reducing sugar. It is a reducing agent. Okay. The reducing agent, the purpose of reducing agent is for the reduction of one product into another product. So that's actually reduction reaction with the help from the reducing agent. So this sugar is a reducing agent. So this is a little bit information. Moreover, after the calculation of the exact amount of glucose present, it becomes easier to prescribe the amount of insulin that must be taken by the patient from the doctors. If we got a problem with the insulin, we got a diabetes, so the amount of concentration of glucose and from this reaction, okay, will be used as a base for the doctor to calculate the amount of insulin in the amount of insulin needed to the diabetes patient. In food chemistry, the level of reducing sugar in the products such as wine, stew, and sugarcane decide their quality. Okay, so because of this reducing sugar, so that is actually the one that I told you about, the technique that we use to determine the sugar content in our blood is based on this Fehling solution. It's based on the sensor. So this will sense the ability of this reducing sugar glucose to reduce something. Okay, and this is how we detect reducing sugar. Okay, there is a this is an example of how we can detect reducing sugar. We have 1 ml of Fehling A, B, 2 to 3 drops of sample, and we put it in, we heat. We got a negative. If there is no reducing sugar, so it will remain as a blue solution. And if there is a reducing sugar, okay, glucose inside the sample, it will change to the red precipitate. Okay, so blue meaning that is a negative. So there are absence of aldehyde and alpha hydroxy ketone. And if there is a positive, if the color change to brick red, the red color, so it is positive. So meaning that the sample contain aldehyde and also alpha hydroxy ketone here. Okay, so this is how we can detect reducing sugar lah, especially glucose. Glucose is a reducing sugar. Okay. So how do we know it's a reducing or non-reducing sugar? You can see maltose, it has a free OH. Okay, lactose, it has a free OH, but unlike this sucrose, and at this position lah, at carbon number 1 here. So this is what we call it as a free OH lah, because it will produce a glucose, and this glucose will form the punya open chain, and this open chain can be oxidized, okay, into something, and we know that, and then it will be reduced to it will call it as a reducing sugar. Okay, so these are the reducing sugar: glucose, fructose, glyceraldehyde, lactose, arabinose, and maltose, and except for the sucrose. Okay, Okay, any question before I come up with the active learning session break? 3:50 now. Is there any question? Can put it in the chat. Okay, so you have learned about disaccharide, and disaccharide there is a maltose, sucrose, and also lactose. And you need to remember the lactose is composed of glucose and galactose. Sucrose is glucose and also fructose. Maltose is come from 2 molecules of glucose. Okay, this is what you have learned today. And then you learn about the reducing sugar. This is also very important. Okay, once you understand that the reduction reaction here, actually I like to have a whiteboard so that I can come up with the reaction, but it's hard to explain. But I think you can understand this. Okay, and then relate to the Fehling test here. Okay, Fehling test here is a reagent lah. Okay, to detect what we call this sugar in our blood. Okay, any question? So far clear, thanks. Okay, can I see some reaction here from you? Okay, thanks. So as usual, it's an active learning. So you can see here. It's a simple question here. Look at this picture. And look at this picture. We have a glucose. It's the color is red. Fructose, the color is red. Saccharose. Saccharose ni is one of the disaccharide. Saccharose is one of the what do you call this? The monosaccharide lah. Okay. And then why do you think the colors are different? I want you to find out, okay, and then I want to you put it in e-learning. Okay, put it in e-learning, put it in Facebook group. Okay, the comment in the Facebook group. Why the colors are different? Okay, simple, fun. Saccharose ni, I want you to Google it. What is the saccharose? Okay, you have glucose, fructose, and also saccharose. Okay, so now you have learned number one here, learn and understand classification in the carbohydrate. So today you learn about the monosaccharide, disaccharide, oligosaccharide, and also polysaccharide. You know about the classification of the carbohydrate. And you understand basic principle of monosaccharide and also disaccharide. And hopefully that you can explain the difference among the monosaccharide and also disaccharide, and demonstrate understanding on example of the type of carbohydrate. So I think with that, thank you very much.